The seminar series aims to increase awareness and collaboration between the two schools, and we select a speaker who satisfies the working in the sort of overlap area between fisheries and oceanography, is involved in fisheries management, and has a substantial impact on the field. Before I introduce Kevin formally, I want to thank the organizing committee, which consisted of myself. I'm not thanking myself. Lawrence Hauser from SAFS, Kendall Valentine from Oceanography, and Bill Kopp, one of the UW affiliate faculty, who can't make it today because he is in Copenhagen chairing the IC's whatever committee he's on. But he does say he would like to be here. So I want to introduce Kevin. Kevin obtained his BA from Rutgers and his PhD from VIMS. He joined NOAA, the Beaufort Lab in, am I allowed to say 1982? Sure. I guess I've just said it, right? And he joined actually before he finished his PhD, so something that a lot of our grad students know all about. And at that time, he was working on the distribution relationships between menhaden and plankton-related gradients. Following his PhD, I have to write this down because Kevin's moved around. He moved first to the Sandy Hook Laboratory, where he joined the Northeast Fisheries Science Center. There he worked on bluefin. The next move was to Woods Hole, and that was then followed by some time at the University of Massachusetts, where he was the director of the Cooperative Marine Education and Research Program. In 2008, he then moved to Narragansett in Rhode Island, where he is a senior scientist today. During his career, he's mentored a whole bunch of graduate students, postdocs, and he's received numerous awards, many of which I'm not going to go into. Apart from the menhaden and the bluefish, Kevin studied, again, a whole horde of stuff, which I think is going to really be brought to the fore as we talk and hear from him today. Species as broad as herrings, sea herring surgeons, eels, cod, salmon, and his published research is sufficiently vast as well, and covers topics in ecology, fisheries science, recruitment processes, etc. In addition to his research side, he's been involved in IC's many scientific working groups, and from the management side, he is a member of the Scientific and Statistical Committee of the New England Fisheries Management Council, so directly involved in basically translation of science into management. And with that, I will leave it over to Kevin. Please give me a hand and introduce.
Wondering how you all started. I realized I sent my CD, and so you went to my CD.
I did indeed.
He's my wife. It is great to be here. Thank you very much for having me. I don't think it's too much to say, but you may have heard from others. It's great to be here in person as opposed to being here on Zoom, and so that's really quite wonderful. Thank you to the departments of Fisheries and Oceanography for having me as part of this series. Really my honor. And also thanks to Lawrence, excuse me, Andre and Bill for sort of getting me organized today. So my title is a series of keywords, and what it reflects is I'm going to talk about what's been on my desk as of late. And so it's a broad spectrum of studies, and I hope you find it of interest. And so before leaving the title slide, I would like to acknowledge my co-authors, really a great group of scientists I've had the pleasure to work with. They make the work fun and interesting, and couldn't do it without them. So the eyes are the windows to the soul. The origin of this quote is not very clear. It's an ancient quote. It's been attributed to authors like Shakespeare and Cicero and Da Vinci. But I have a more modern take on it. I think that Amazon provides the window to the soul. And so through the course of my talk, I'll share with you some of my Amazon purchases as a way for you to get a little Get to know me a little bit better. So oh boy, it's cut off. So the first three.
You can click that X and then we'll get rid of that. If you go to the computer, click the little X. it'll get rid of that.
I click the X. There we go.
You may
So
also
the-
click your slides so the clicker works again.
So the first three keywords are climate change, temperature, and phytoplankton blooms. And I'll talk about these three studies. The first of which is asymmetry in the rate of warming and phenology of phytoplankton blooms on the Northeast Shelf, which just came out in NICE's Journal of Marine Science. So what we noticed some years ago is that on the Northeast Shelf, we're seeing a sort of an asymmetry in the way the system is warming. And this is illustrated by this paper, figure from this paper that John Hare and I did back in 2007, where you're looking at normalized minima and maxima for 7 index areas in the Northeast Shelf. And the point that it's illustrated for you, if you look at the blue lines, which are the minima, they're not really raised, they're not really going up in the most contemporary period. Where you see this divergence amongst the maxima where they're rising quite rapidly. So what we're seeing is that each year there's a pretty dramatic excursion in temperature that occurs in the warm part of the year versus the cold part of the year. So in this study this new study, we took a more detailed look at this warming and this asymmetry to try to understand it in the context of whether there's any change in phytoplankton bloom phenology. So the data, the SST data was from the OI SST data set, which you may be familiar with. So we extracted daily SST data for 3 index areas or 3 ecoregions on the Northeast Shelf, Gulf of Maine, Georges Bank, and Mid-Atlantic Bight. In addition to looking at trend in the temperature, we also looked at what we call seasonal thermal transition indices. And simply what we do is we calculate a mean for the respective area, and then determine when during the year temperature transits through that mean as an indication of the first transition in spring, when that thermal transition is occurring, and then the transition in fall. The chlorophyll data is based on satellite remote sensing data. We extracted 8-day time step data, again for the 3 ecoregions, and examines bloom start and duration to assess bloom phenology. I utilize what I call a change point algorithm to determine when a bloom is occurring. In which case, we have to find the onset of a bloom as a positive change point, and the end of a bloom, which is a negative change point, to bracket when the bloom occurs. If I can't find those 2 change points, we don't score the bloom as occurring. But that yields data on when the bloom starts and the duration of bloom. The search for a bloom occurs over what I call a search window, or 1/2 of a year. To find that bloom event. So trend in SST and bloom statistics were determined using Mann-Campbell tests. Here's the chlorophyll concentration by the 3 ecoregions, the top panel being Georges Bank, Gulf of Maine, and the bottom being Mid-Atlantic Bight. So you immediately notice similarities in the pattern for Georges Bank and the Gulf of Maine. In both systems, there's a spring bloom in April. And then also in both systems, there's a tendency for an autumn bloom in October. This is kind of a very unique pattern in the North Atlantic. Most of the North Atlantic is dominated by systems with a single spring bloom. But we're in a latitudinal band that extends across the North Atlantic, where there is sufficient warming and cooling and stratification mixing that occurs in the fall of the year, while there is still sufficient light to stimulate the second bloom. There's a very different pattern in the Mid-Atlantic Bight. In our cold pool, there's a very strong stratification structure that breaks down the fall, which leads to this fall-winter bloom phenomenon. But when it reforms, it sort of stunts the development of spring. So part of the reason I started with the chlorophyll data, you'll notice these markers when these blooms tend to occur, they'll carry over into the next figure. So the top panels are the mean SST by day of the year for Georges Bank, Gulf of Maine, and Mid-Atlantic Bight. No surprises here, it's cold in the winter and hot in the summer. But the adjoining panels in the bottom are SST trend by day of the year. again arrayed by Georges Bank, Gulf of Maine, and Mid-Atlantic Bight, where each trend value for each day is marked with a cross, and the black crosses versus the gray crosses are significant trends. And carrying over from the previous figure, gray bars mark when the spring, autumn, and winter blooms tend to occur. So if you visually scan down when these are occurring, you'll notice the pattern. The spring bloom in both Gulf of Maine and Georges Bank is occurring when there's really not a well trend in daily temperature, whereas the autumn bloom is occurring when there isn't a strong trend in temperature. Likewise, the fall bloom, fall winter bloom in the Mid-Atlantic Bight is not associated with trends in daily temperature. Here are the thermal transition data for the spring and fall generated by Georges Bank, Gulf of Maine, and Mid-Atlantic Bight, where there is a red line that indicates that there's a significant trend. So for the spring data, the only significant trend is realized in the Gulf of Maine. With the autumn transition, there's a significant trend in autumn transition in all the ecoregions. So there's more of an evidence of thermal transition effect occurring in the fall. Here are the bloom data for the Georges Bank ecoregion. So we have the spring data in the upper panels and the autumn data in the bottom panels for start day of the year and duration of the bloom itself. So the spring data, there are no lines, so there are no significant trends in start day or duration in the spring bloom on Georges Bank. However, in autumn, there are red lines indicating that these are significant trends. So there's a shift to slightly later blooms, autumn blooms on Georges Bank and shorter duration blooms. We can blow through the Gulf of Maine data very quickly. There are no lines. There are no trends in between. And likewise, there are no trends in the Mid-Atlantic Bight. So we can summarize this first study by acknowledging that there's this asymmetry or seasonality in the way the ecosystem is warming in this part of the North Atlantic. But equally, we also see that there's very little evidence of any kind of phenology effect to that transitional effect, thermal effects in spring, whereas there's more profound effect in the fall. And then also that bloom phenology is the analysis that suggests there's really no effect on the time and duration of spring blooms, where there's something of an effect on this late season autumn, at least in one area. So I bought this Douglas Gigi black poodle dog, and I bought it for our dog Vanta. There was a period of confusion between the two. Vanta was really not sure what to do. She got over it, she chewed Gigi's head off, and Gigi had to go. The second study is patterns in the warm and cool, cool and warm sea surface temperature in the North Atlantic, the dynamic response to the spring phytoplankton bloom. This one is still in review. Hopefully it's doing well. So building on this previous analysis, I was curious as to whether this is a more generalized pattern across the North Atlantic. And so utilizing the study grid of 2-degree boxes that are shown in gray across the North Atlantic, we did the same sort of analysis that we did before with the temperature data and the chlorophyll data. So as mentioned here, the analysis was done the same way, but limiting it just to the spring bloom is the dominant pattern in the basin. SST again is from the OI SST data sets, same as the previous study. SST and thermal transition indices are the same, but we extended the methodology with the temperature by utilizing the method from Lee et al. 2021, this sort of quantile method to identify extreme warm and cool conditions. And what is done is to characterize the mean temperature of the coolest 10% quantile as an index of cool temperature, and then the mean of the 10%, the 90% quantile as an index of warm temperature. And that's illustrated in this table where we have a time series of 20 observations. You see some patterning to the data. Essentially what you're doing is sorting it. And then for the first quantile with the cool temperatures, you're just taking the mean of those temperatures that fall in that quantile. Likewise, for the warm temperatures, the mean of those temperatures that fall in that quantile. In addition to that, you go to a location and you compute the long-term mean quantile cutoffs. With those temperatures in mind, you go back to the annual data and you calculate days cool SST and days warm SST as those days that exceed or are less than those cutoffs. So it becomes a bulk measure of the number of days that are cool temperature to warm temperature. So here are the SST trends. In panel A is the trend in mean annual SST. Where you see color, there's a significant trend. Where you see white, there's no trend. And what you see is around the basin, there's red scores, so they're indicating that temperature is increasing annually throughout the basin, with the exception of what the oceanographers call this warming hole in the center part of the basin. We look at the trend in mean and spring transition day. We'll see around the basin there are no significant trends. There is something going on in that warming hole. But moving on to the more interesting part of first the cool days, cool SST. We see that there are significant trends around the basins with the exception of the northwest Atlantic. That's a good thing because that makes it consistent with what I showed you in the previous study. We also see trends in the number of cool days. It's blue, so they're declining. And they're declining everywhere except in the northwest Atlantic. Then repeat the exercise with warm SST and days warm SST. We see significant trends in the warm part of the year, or the warm SSTs, in all of the North Atlantic except the southeast. And likewise, an absence of the trends in warm days. What I find so fascinating about this is that it gives you a variety of patterns of warming throughout the basin. Everywhere it's warming. In some areas it's warming symmetrically in both seasons. Some areas warming in warm seasons, some areas are warming in cool seasons. Then we can line this up to the bloom trends. So the figure A is trends in bloom start. There are not many significant trends. They are less than 6% of the points, so that it really doesn't elevate it above random noise. So we conclude in the North Atlantic Basin, there's no coherent trend to bloom start day. I would say the exact same words about bloom duration. There's no coherent trend. So the North Atlantic is warming, however, it is warming in different patterns of asymmetry across the basin. And as I mentioned, it provides a natural experiment as to whether these warming patterns have an effect on the spring bloom phenology. No evidence of any change in spring bloom phenology. So it leads to a natural question: what will be our expectations for bloom phenology going forward? We went through a period, a multi-decadal period of climate change that we would expect or anticipate to be pretty much the same that we'll experience in the coming decades. Does that mean that the spring bloom is resilient to these changes? I bought a Springbok 500-piece jigsaw puzzle, Morning Serenade. It was the pandemic. So this one's in preparation. Regime changes in the Northwest Atlantic, sea surface temperature revealed using a quantile approach. So this started as a data exploration, but I'm really kind of excited about the results that came out of it. So looking at the SST on the Northeast Shelf, but also looking at the SST and the associated slow sea. That'll be clear in a moment why we want to do that. Same SST data as before, same trend methods as before, but we're also going to look at change points using stars, which I would wager is very familiar to you guys. So it's an extension of this 10% quantile method, but instead of using 10% quantiles, we use 25% quantiles or quartiles. And instead of just concentrating on the warmest and coolest temperatures, we're looking at them as bulk measures of exposure. So again, this illustrating simply a time series of 12 observations. Now we're sorting into 4 bins, and we're going to utilize all 4 temperatures to try to understand what's going on with temperature change. So the study area is shown in this figure, and where we've been always so shelf-centric in previous analyses, I've extended that well beyond the shelf by the study grid shown in black dots. In addition to that, the change point analysis was predicated on these 5 cumulative index areas, which are quite frankly selected by law. But they are Gulf of Maine, Georgia's Bank, Mid-Atlantic Bight, but then also the northern slope C and the southern slope C marked by purple and mustard. It'll become obvious why I've selected those in just a minute. But first, let's take a step back and talk a little bit about the oceanography of the Northeast Shelf. The Northeast Shelf is affected by the same radiant and atmospheric warming and cooling effects elsewhere. And so that sort of puts a global expectation of about 0.1 to 0.2 degrees per decade change in temperature that you expect to see. You will see that the Northeast Shelf is doing some other things, and what those other things are is a result of warming that's caused by transport of different water masses into the system. I selected this figure because it shows the bathymetry of the Gulf of Maine, in particular the Northeast Channel. And what is so interesting about this system is that it represents the confluence of two major current systems. The Labrador Current, shown in blue, and then warm slope water associated with the Gulf Stream in red. And what has been very typical over the years is that the Labrador Current has played a dominant role in advection of Labrador Current water in the Northeast Channel into the Gulf of Maine and onto the shelf itself. And what that does is it transports relatively cool fresh water that has pretty good nutrients. On the other hand, warm slope water tends to be warm and salty and often can be nutrient poor, or sometimes stoichiometry poor and lack silica. So what has happened as of late is that the Labrador current water has been replaced by more warm slope water due to changes in basin scale circulation. So we're getting an influx of more of this warmer water. And the changes have been dramatic. So this is trending the mean annual SST. So you'll notice in the Gulf of Maine, we're seeing changes of a half a degree per decade. That's off the charts. That's really a lot of change. And over the study period of 4 decades, matching the SST data, and that's 2 degree shift in the thermal conditions. The rest of the shelf is more like 0.2 degrees or so per decade. However, when we go offshore, we see patterns of change that are approaching 1 degree per decade. That's obviously not warming by normal processes. This is just warmer water now displacing cooler water. And we see 2 pretty intensive locations for that, where that warming. Here are the trends in SST by quantile. So the cold water quantile is really not, you don't see a coherent pattern trend, which is a part reflecting a lot of noise in the data as well. But we see trends start to emerge in the second, especially the third quantile, where we're seeing tremendous change, now approaching a degree per decade in these offshore slope sea areas. And then finally, the last quantile, where we see peak trends that are in excess of a degree per decade. That's equating to a 5 degree shift over the study period. That's amazing. And though they're off shelf, a lot of our resources use off shelf water that's fluid in that. Here are the changes in quantile days. So we see blue tones in the cool water, meaning that there's a loss of these cool days. And you see numbers that are 15 or so. There's sort of a zero-sum game in these intermediate levels, but then there's a gain of days in the warm end. And these are numbering up over 25 days of additional warming conditions. Here are the results of the time series analysis, which I promise I won't go into detail. But just to give you a sort of an overall feel for them, they're arranged by parameter which we are working with, and then those ecoregions. And as I skip through these slides very quickly, the thing that you should do is first, the red line shows the stars mean, so you see where the transitions are being identified in the data. But I've also marked 2012, which is the warmest year on record for our ecosystem. And you'll see that it's a pretty significant watershed. But in addition to that, as I flip through these very quickly, you'll notice that so many of these change points are near 2012. here are the quantile temperatures, and then the quantile days. So I sort of summarized all these change points into this figure, and I would point out two features that are really kind of interesting. One of these two change points that occurred in 1998, 1999. They are change points that are associated with a change in time, which is kind of interesting. Then, as promised, 2012 was really quite a watershed, but what is also quite interesting about this is all the precursor change points that occurred as a run-up to this condition, especially the ones in 2007, which occurred in the southern slope sea. And we see this as sort of precursor moves of the ocean circulation before it starts to affect the shell, but we're seeing evidence of that change in circulation offshore. So the take-homes from this particular study is a remarkable level of warming due to these affectional water masses. And that this, we think that this method hopefully will stimulate some interest, because we think it sort of really illustrates the potential for exposure to thermal conditions in a lot of ways that other thermal indices don't. The 1999 change points were very interesting because they were also associated with change in development of work rates, changes in zooplankton community structure, and improvement of fish. So even those small change points had meaning. As I said, 2012 was the warmest year on record, and there's a linkage to the basis gas circulation that we start seeing. That goes up in 2007. So before leaving this group of keywords, change point, temperature, and phytoplankton, I would like to put out some questions for you to think about. Is there a need for a comparative look at bloom detection methodology? I think that many of you are probably not familiar with the Hoover blooms with change points, but I think that I would like to bring that to the fore a bit more. Where and under what circumstances is bloom phenology actually shifting? Do we see a role for the synoptic comparative use of quantile characterization of temperature change? Is it a useful indicator of thermal stress? And this would be in the context of other indicators like wave heat analysis. And I just do this on because this is something I'm working on recently. Our freshwater nutrient sources playing a significant role in continental shelf productivity, and if you're curious about that content. So I bought some Earth Re's laundry detergent sheets, fresh scent, no plastic jug. Because they're really good choice. Plastic jug with so much more. So the next two papers relate to what's going on with the Northeast Shelf and wind energy development. And the first one touches a little bit upon forage fish resources as well. So this first paper is forage fish species prefer habitat within designated offshore wind energy areas in the U.S. Northeast Shelf ecosystem. This one just came out in Union Coastal Fisheries. But I'll start off by talking a little bit about what wind energy is starting to look like, because the towers are going into the water as we speak in the Northeast US. So this is a map of Ocean Wind 1 off of the New Jersey coast. And what is kind of interesting about it is that the blue dots mark where the wind towers are located. And they tend to be spaced by anywhere from 0.8 to 1 nautical mile. What else is interesting is that the blue lines represent the wiring between towers. And I don't know about you, but I naively thought, oh, well, they'll have them all going the same way. Not so much. It's really quite a tangle of how they have the wiring going into two different X-Y directions. So having said that, and we'll return to this topic in a few slides, our boats are not going in there. Fellow boats are not going in there to work. And you have to also wonder what fishing gears would be able to work in this area as well. So the first paper I want to talk about is the use of random forest species distribution models to assess the potential impacts of wind energy development. First, let me talk about this family of models that we've developed in a more general sense, because I use them in a number of other papers as well. So first is that So as I said, we'll return to this in a moment. So the estimates for habitat, both occupancy habitat and biomass habitat, are performed over this grid that's shown in blue for all these models of what we're talking about. And I have two versions of these models, which are based on random forest selection of variables from a host of physical and biological explanatory variables. I have an early version and I have a late version. The early version is based on a shorter time series that began in 1992, when I was trying to include salinity as a dynamic variable. I also was pretty conservative in model selection in terms of which models to include, and only utilize 60 species of the species that are on the shelf. This later version has a longer time series beginning in 1976, where I dropped the dynamic use of salinity, but I also am more inclusive in model selection, and I model 177 taxa random forest models. So this is the map of the, again, it's in a different color, of the output from our species distribution model, but superimposed on that in green is a recent depiction of where the lease areas and interested coal areas for wind area or for wind energy are located on the northeast shelf. As you'll see in a couple of slides, this has already changed. It virtually changes day to day where the boundaries are located. But for the purposes of this study, it's adequate to get a sense as to what's going on. I'm starting with the latter version, the 177 species. And what we did is one aspect of the study was to look at core habitat, or what I call core habitat versus overall habitat, which is demarcated by the 20% quantile of the habitat scores versus the 80% quantile. And this is illustrated for butterfish in this figure. And so where you see this dark cyan, that's the core habitat where we have the highest probability of occurring, versus the overall habitat in lighter cyan, where they have a likelihood of occurring, but much lower. And what we did is we assessed the relative use of release areas by all species, but with a focus on forage taxa, by looking at the ratio of core to overall habitat in the lease areas versus the core to overall habitat throughout the ecosystem. So a species with a high ratio is one that likely is utilizing the wind areas to a higher degree. And what was kind of interesting about this study is that what we identified as forage taxa tended to show relatively high utilization of the areas. I thought that was kind of interesting, which I've already summarized here. Wasn't we cracked? They were full of fiber notes, and you can enjoy them any time. I bought them as part of my eternal quest to find a replacement for stone. Yes. Which the manufacturer got rid of, and I can no longer enjoy wine machine. So the second study is in review. Some observations on spatial requirements of fisheries sampling in a world with energy infrastructure. This is a picture of our survey strata for the bottom troll survey. We do a number of surveys in the Northeast, many of which are going to be impacted by the installation of wind energy infrastructure. In other words, we don't have the ability to carry out the study design because station selection will be interrupted by the infrastructure. So how do we adapt these surveys to this new reality? One thing that we thought we would look at is spatial correlation. So here's the map of current wind lease areas in blue and interest areas in red. As I told you, the map is changing constantly. The interest areas tend to be very large. So we're starting off with consideration of the entire Gulf of Maine for development of wind. But I can rest assured that it will be smaller lease areas that are actually licensed for operators. And you'll notice that the blue areas are leased and under development, and they tend to be smaller. So in addition to estimating the typical spatial dimensions of lease areas, estimated the spatial correlation of species by calculating range estimates for variograms by year and by season and species. We limited the analysis to only species that utilize our bottom troll survey for assessment purposes. make it a bit more relevant. So here are the spatial dimensions of the wind lease areas, and as you can see, they tend to average about 300 or so square kilometers per lease area. And though there's no official policy, there tends to be a navigable edge to all these areas. In other words, an operator does not have to pass through other people's lease areas in order to get to their wind fields. That's important from a sampling point of view, because that means there is potentially an area where you can operate a trawl without being in the lease area. Then, for each of these lease areas, I calculated all combination of core distances, to sort of come up with a sort of an average dimension to them. And so I put forth these three measures, 8, 14, and 21 kilometers, as an indication of sort of the range of values of how close you would need to be to a lease area with a trawl. Here are the spring variogram data, shown as box plots with the 10 to 90% quantiles in light red, 20 to 75 in dark red, and medians in black lines. And sorry, but these are species codes, and I only bring a couple names for cod and shad. But the thing about it is that if you look at where the dashed lines are, 8, 14, and 21, most of the data, most of the variogram data would suggest that the spatial correlation far exceeds the dimensions of the lease areas. And also keeping in mind that this is a log scale, so it's quite a bit more than it looks graphical. But this is kind of encouraging, because what that does, well, this is a lot of data, pretty much the same story. But what that does is suggest that we can have sampling designs based on fixed stations or hybrid fixed randomized stations that potentially could be relevant to the lease areas without having to go into them. And that's the conclusion. So looking forward, wind energy has the potential of completely reshaping our continental shelves and beyond. But not only that, reshaping the monitoring enterprises we have to keep track of what's going on. And it is coming. So go on. Also, it is also intrigued by this core to overall habitat phenomenon. Again, that picture, just let me know. I bought a schematic pink noise machine. So I get white noise and pink noise, but what's up with brown noise? Hopefully, there's no brown noise in it. No Mythbusters fans. So the last one I want to talk about are the keywords habitat, biomass, and growth. So this first paper I'll talk about came out in 2020, a few years ago. Changes in higher trophic level productivity, diversity, and rich space in a rapidly warming continental shelf ecosystem. I mean, as you are well aware, climate change is transforming ecosystems. This was, I thought, was a really striking data set from Hiddick and Hofstede for the North Sea, showing change in species richness by year. This tremendous change in the species composition in a relatively short time period. But these processes are pretty widely spread amongst not only distributional changes and species richness changes, but also changes that would occur affect metabolism and growth, potentially growth. But I like to think of them all sort of under an umbrella of tropicalization of these ecosystems. That it's not only limited to the full transformation of an ecosystem to look like something that was at lower latitudes, but it's also these subtle changes to how the system is organized and function as well. So again, these random forest models have reared their ugly head again, this time with the early version, 60 species shorter time series. This is an example of what the winter flounder occurrence habitat looks like. And what we did is we examined the trends in habitat area by species over time to ask whether the occupation footprint is changing. This is what the data looks like grouped by functional, the data trends look like by functional groups for benthivores, demersal piscivores, pelagic piscivores, and planktivores. And you'll notice that by and large, they're all upwardly trending. So these data suggested to us that the occupation footprint of many taxa across the Northeast Shelf is changing, and they are occupying wider areas. In addition to that, we looked at trends in overlap or interaction area between species pairs. This is to give an idea of whether there's potentially more species interaction or competition. So in these figures for A and D for spring and autumn, the blue line, the blue dots indicate that that species pairing had a trend in interaction area that's positive or increasing versus red and negative. And you see the biomolecules that are showing increasing trends, so that there's more spatial overlap between species. So it's an indication that species occurrence footprints are increasing, as I mentioned, and that as a consequence, there's more hope, habitat overlap and potentially more competition. I bought a red Vex pace camera bead bracelet, which is really great for counting laps while swimming. Pop quiz: what is the only pack quality on a school bathroom? So this next paper, the relationship between habitat area and biomass of fish and mackerel invertebrates on the northeast shelf, just came out in Marine and Coastal Fisheries. So I got a fair amount of criticism about the first paper that people were saying, oh, what you're seeing is driven by one species, we're going to break it out sufficiently to be convincing. So this time, using the random forest SDM ladder version, more species, longer time series, looked at trends in habitat, but then also looked at swept area estimates of biomass based on SDM post-stratification. What the hell is that? So if you look at this continuous depiction of habitat for a taxa, I think it's winter flounder again. This next figure is the habitat is integralized over 10 intervals. You can see that pixelation a little bit. But the lowest interval becomes a stratum, as marked in this figure. And the highest interval, the most desirable habitat, is also marked. And so what we do is we assign the relevant codes for those habitats to those strata and compute a swept area abundance for those areas. And then sum it over the entire ecosystem. Come up with an ecosystem-wide estimate of biomass. In addition to a trend test, we also looked at a t-test to test whether the trend by comparing it earlier than parts of the time series. So here's a summary of the data for habitat area and swept area estimates of biomass for spring and for autumn. Everything's going up. So we took this as an indication of, again, another way of looking at this increase in footprint or distributional space and an increase in biomass. These are tree maps of significant trends of biomass, blue being positive trends and red being negative for the spring, and then fall, the lower panels, the trend and then difference indicator. And the point here is most of them are blue, so most of them are trending upwards. There are some big players, but the point is that there are also many other small players as well. So for bottom trawl species in the survey, based on a grouping that represents nearly all the biomass captured by the survey, that's kind of an important point. So we think about the compartment of biomass that the survey sees. I'm now accounting for 99% of that biomass looking at these trends. Nearly all the biomass capture has increased, and so has the biomass associated with it. Both habitat and biomass are increasing, and for a majority of species as well, addressing that other point. But it leads to this, what I'd like to call the habitat area biomass hypothesis. With increased temperature, habitats have expanded. In other words, you have more areas where there's tolerable temperatures for species. And those expanded habitats now are in a position to support larger populations, but also causing more species overlap and competition. So where do we go with that? First, let you know that I bought a copy of Amazing Stupid Mad. I was looking for the Metamorphosis by Kafka. I couldn't find it. I figured this was a similar genre. So this last paper is interviewed, change in body size and rapidly warming marine ecosystem consequences of tropicalization. So you notice there's a little bit of a regression here trying to take steps. Latter version of the SDMs, estimates of biomass, and now also estimates of abundance. provide estimates of individual size. So if you know the abundance and biomass, you know, this rough measure of what the body size is per individual. Also looked at a length indicator based on raised estimates of what's seen by the survey. So it's sort of independent of the other estimation techniques. In addition to looking at all the taxa, we also developed it for an index of 51 of the species. And what's special about them is they occur in both spring and fall surveys, and they have virtually no missing values in the time survey. So we can view those as well estimated by the survey, giving sort of an apples to apples comparison as we look at it over time. First, a disclaimer. As soon as you talk about change in size, there's a million different hypotheses that emerge. Fisheries, removal, genetics, oxygen, primary production, among others. We're very aware of that. So we're putting forth a hypothesis and evidence to support it one way or the other. And this, we acknowledge that we have an incomplete test of all things that are possible. In addition to that, we considered the catch for the northeast shelf large meat ecosystem that we see around us, less taxa that are associated with estuary, like oysters and things like that. And so we're partially looking at fisheries removal. Not really a tremendous job of it, but a little bit. And I would also bring forward now, while I have before I look at the actual data from the analysis, chlorophyll and by association primary production has declined in this ecosystem. And I say that with some confidence because not only do I see it in the remote sensing data, but inside you data supports that contention as well. It's actually measured with the type of stuff that has been a decline in chlorophyll, if any. So here's the data from spring for all in common species. All species are in black, common are in the red traces. Where there is a dashed line that indicates a significant trend. Here are the trends in abundance and biomass, and the trends in weight per individual, and the trends in length per individual. So for whatever it's worth, from the spring, we have evidence of an increasing biomass and abundance. They're not scaling by the cube, so the animals would appear to be getting smaller, and they're also getting shorter. Here's the fall data. Pretty much the same story. Here are the results from the catch data for the Northeast Shelf. For the common pool species and all taxa, there's no line, so there's no trend. So this is a really trust me type of analysis that suggests that catch is not really, catch is a lower likelihood explanatory variable for what we're seeing. So there's evidence of a concomitant expansion of biomass and abundance. This expansion was not related to the cube size at the time. Consistent with this previous hypothesis that I mentioned, this idea of area biomass, that with increased temperature, habitats have expanded, which is supporting larger populations, but also causing more overlapping competition, and hence that's also causing a decline in size. Primary production has not increased to support increased biomass. Increases biomass increases around 50%. So what does that suggest about primary production and its role? Were previous biomass levels not utilizing the full availability of primary production? I don't interpret that. So again, looking forward to this grouping of habitat biomass growth, is habitat more important than primary production in influencing energy utilization efficiency? I don't have an answer. I think it's kind of interesting. Have we considered energy flow in the proper context of habitat? And what can we learn about energy flow, carbon sequestration, and production with system comparisons that account for habitat? So I bought these things, deep blue fins, because after this, I'm going online. And finally, let's take a look back at Kevin's soul. He has a certain indifference to the fate of stuff down there. We'll do a jigsaw puzzle on duress. lazy, but also is concerned about festivals. Loves good cracker, perhaps has a phobia about brown noise, needs remedial help in counting, has an egalitarian view of literature and existing division. Thank you very much.
Got some time for questions. Firstly, thank you, Kevin, for an amazing tour through everything that we were after. We covered everything from lower trophic levels to wind farms to fisheries yields. You know, if we don't come up each of us with 10 new questions, we're not paying attention. I also at this point like to thank Kevin. Maybe I set the bar a little high in terms of how many meetings you have today. I just realized this is his 10th hour of interactions with us. We started at 8 o'clock this morning at Portage Bay, and he's still more or less going.
I thoroughly enjoyed all my conversations with the people I have a chance to meet today and look forward to tomorrow.
And there's tomorrow, and there's beer after this, and yeah, it'll continue. But questions? Yeah, Trevor.
So I was looking at happening the wind farm areas, and it occurs to me that there's no fishing inside those wind farm areas. They're de facto MPAs. Is this like a 2-for-1 where you can say you're protecting 30% of the area with MPA, and you're also producing wind energy, or
So the wind industry has declared that anyone can go in there and do whatever they want. You can't touch their power, but they're presenting themselves as not an impediment to any kind of fisheries operation. There's a harsh reality of not being able to do certain years in them.
Yeah.
They will be able to do other years. And I haven't kept up with it, but I do know in Europe, the first settled pontoon, they were real ideas. They were not allowing activities. Europe is in the process of really expanding the footprint for their wind energy area. So they're going to have to come to this with the possession as well. So we're speculating with rates that we know. I mean, I can foresee a shift in years. I can see pots being fished there, but fuel nets. I have a hard time seeing them being great enough to try to do an active duty.
I had a question from the beginning of your talk about the fall bloom. What's triggering that? That's the next.
So as I mentioned, what's happening in this particular latitude level, and we see it all across the North Atlantic. It's cooling enough for stratification to break down, while there's still enough sun in the sky to stimulate it. You get that mixing, nutrients come up to the thermocline, it's broken down, but it's still sunny.
So you're getting mixed layer deep enough to bring nutrients into the upper ocean.
The mixed layer is shallow, it's shoal. In other words, it sets up in the summer.
Okay.
As it's cooling, it's breaking down, so nutrients can come up and be able to.
In freedom?
And just a comment, in freshwater systems, you often get
For the same reason.
my question is about the fish getting smaller. Are they younger? Or are they growing more slowly per age, which might mean something about how you produce more or you've got more biomass, but you've got smaller fish.
Yes. I'm being facetious because it is really tough to sort it all out. So in the previous paper, we looked at growth data on individuals that were collected in the survey, and we saw evidence of a decline. But as you start to add aged fish into the equation, I cautiously believe they are doing the same thing, but someone has to start doing the meta-analysis of all the assessment data to start to put that together in a more disciplined fashion. I don't know if that answers your question. So a lot of it is just not synoptically addressed yet.
Well, I'm just thinking of the Alaskan situation where the argument is that because the nets are more efficient in catching large fish,
Yeah.
the ones that are left to reproduce are smaller. So, I mean, there may be many mechanisms of fish getting smaller.
If you remember in my slide, I cowardly listed all those and say, we have to pay attention to them, you know. But at the same time, there's other things I know about. So for example, we do some content. Though I don't have access to a complete synoptic view of all some content consumption rates. For many species, consumption is declining. That would be the system of that hypothesis. There's more. That's
So I have a question. Oh, actually,
Following up to the wind energy question, are you aware of any, in terms of the wind farms and environmental impact assessments, is there any official thing at the federal level that dictates sort of how environmental impact assessments be conducted or best practices? We're represented instrument manufacturers, and we hear a bunch of different.
There are many syntheses of the science, either published or being published. I mean, groups are scrambling to sort of like put all of this in some sort of codified fashion. You know, I would just say that there's two levels of things in the ethic. One is benthic modification associated with power. If you just keep in mind, those are impacts that are like tens of meters away from the tower. So towers are spaced by a kilometer, you know, it doesn't take much to do that. A wind field will modify a single-digit % of the benthos. The other one that is more frightening, the more intriguing, is that you have wind turbulence, you have turbulence in water column caused by towers. And they can cause a mesoscale effect on turbidity, temperature distribution, stratification, which could extend well beyond the wind fields themselves. So I think, you know, we're very concerned about both. Those two spatial states.
Jim?
Yeah, and Kevin, thanks for coming and giving a wonderful talk. I was curious if you could kind of lay out a little bit more like the essential fish habitat or kind of fish habitat mapping implications of some of this work. I mean, it seems like you're showing changes in distribution and area occupied now. I'm curious how the Northeast region has sort of responded to that, either in the SSC or the council.
It's like, I was going to say cottage industry, but it's actually a major industry developing SDMs.
Yeah.
There are many groups that are developing SDMs. There's an official one associated with councils that are doing related but different analyses. I'm quite happy to see those be more official data streaming assessments. I presented these data academic, you know, in academic journals and peer-reviewed journals. I've also presented these data in the context of assessment working groups. So it's being used in a number of different ways. There's a number of different players, and that answer your question?
Yeah, and I'm curious if I'll follow up later. Yeah, thank you.
Yeah, I mean, I got hordes of questions, but one I'm really interested in is you're on the SSC of the Northeast Council. And the Northeast Council, for those of us who live in the West Coast, is sort of, if it goes wrong in your region, right? And so maybe you're the canary in the coal mine for the problems that the rest of us are going to face. How, in a management context, is these sort of changes to the baseline really entering into the management process, or is it really entering into the sort of scientific journals but not actually impacting how councils actually make decisions? Where in the process does this come into being, and what's our future?
You say baseline, you say ecological change.
Well, the baseline is sort of, this is an overexploited ecosystem that we've been trying to recover for decades, but now we've got all these changes. And, you know, the baseline of, we've got an overexploited ecosystem that we're building, this just adds a whole dimension to it. And I'm trying to work out how this stuff feeds into the decision-making process, if at all. Apart from retrospective.
We have, in the Northeast, we have two councils, the Mid-Atlantic Council in the Northeast. So what we began a number of years ago is to directly go to the council to provide them reports on the state of the ecosystem. And our intent here was, if nothing more, than to give them the vocabulary and perspective on what this data might look like and how it's affecting the resources they're managing. The councils do not like to seek information being put on the floor at the meetings themselves. You know, they want to see it come up through the peer review process from working groups, PDPs to SSCs to the council. And so that process is being facilitated by stuff that you guys saw, ESPs and stuff. So we're adopting that framework and format to try to more aggressive, more truthfully bring in this information into the assessment process, so that it finds its way of being either directly incorporated into the models that we use to estimate catch levels, or it is brought along so that it can be used in a risk assessment context by the groups that provide the rest of the census. And that's starting to occur in the Northwest. Does that answer your
Yeah, very much so. Okay, I think we should thank Kevin again.